still in Chapter 2. I'm on page 39. This is where we're talking about the different kinds of T lymphocytes. Spend a little time. Again, you're learning some of the basics of a language, so the more you understand the terms that are in here, the easier it's going to be later on. T cells are designated because they're born in the bone marrow, but they're actually educated in the thymus gland. They have to learn to recognize self versus non-self. So they learn to bind to cell membrane receptors called MHCs, major histocompatibility complexes. There are two types of MHCs. MHC1 are usually produced on every cell in the body that's nucleated. So there's a few exceptions. Sperm and eggs don't produce MHCs. Red blood cells typically don't produce MHCs. But these T cells are going to have to learn to recognize that. And it's like having a name tag on your outside surface. MHC class 1 tells you what's inside the cell. So you've got empty name tags all over, hundreds of them, but you take little bits of what's inside the cell and put inside the name tag. So you slide liver, you slide spleen, you slide nerve, you slide other proteins into those name tags, and it gives kind of an impression of what's going on inside the cell. Well, if the cell happens to be infected with a virus and viral protein gets inserted into that, then a cytotoxic T cell can come along, recognize the MHC class 1 molecule is self, but it's carrying foreign antigen, and it knows that it's time to kill or knock out the cell because it's carrying something that's foreign that doesn't belong. That's where the education comes in for these T cells. They have to learn to recognize self MHCs, but not overreact to self antigen that's carried inside those name tags. Now, the class 2 MHC molecules come from professional antigen presenting cells like macrophages and basophils, and even you can find them on the surface of dendritic cells. So what you do is you take these MHC molecules are going to be working with exogenously obtained antigen, where in MHC class 1 molecules typically are going to be expressing endogenously obtained antigen. MHC class 2 is going to be working with exogenously obtained antigen. So a macrophage eats something that's from outside, like a bacterium, chops it into little pieces and puts those pieces on the outer surface of the cell. Okay, you can read through this. I mean, there's a lot of really good information on page 40 in this unit. It takes some time to try to understand it. There are different types of T helper cells. T helper cell type 1 are usually used for intracellular pathogens. T helper cell type 2 are usually used to cause or regulate a response to extracellular pathogens, where there are other types of T cells, like T helper cell type 17, responds to interleukin, which is just a chemical messenger, interleukin 17, and it causes cell-mediated immunity, which means you can recognize specific fungal or bacterial or viral pathogens, and it stimulates a really strong cell response where it goes in and you kill those cells that are infected. This could be very useful in treatment of diseases like HIV in the future. Also, there's another type that we generally don't talk about much, but it's a T regulatory cell, and these are becoming more and more important. They're finding recently that these T regulatory cells are overproduced in some cancers. So if you can go in and specifically shut down those T regulatory cells with cancer, the regular immune system kicks in and you get a cell-mediated response which kills those cancer cell types. And they're using lots of immunological techniques for treatment of cancer and other conditions. And just by regulating those cells that are regulating the response, these are like suppressor cells. A lot of times the T regulatory cells just say, oh, whoa, wait a minute, don't over-respond. Well, if we can selectively turn off certain groups of those T regulatory cells, then we could cure certain types of cancer. And this is really big. I think you're going to see a lot of that happening over the next decade or so. All right, I'll stop and we'll do natural killer cells next.